Hi everyone! In this explainer, we will be examining a study from Nature Medicine that investigates the role of physical activity as a potential modifiable risk factor in the preclinical stages of Alzheimer's disease.
Our focus today is on a longitudinal study that looks at the relationship between physical activity and the progression of Alzheimer's disease pathology. Specifically, we'll be exploring the effects during the preclinical phase, which is the long period before clinical symptoms become apparent.
We'll structure our discussion as follows. First, we'll establish the context of preclinical Alzheimer's disease. Then, we'll review the design of the Harvard Aging Brain Study. From there, we'll analyze the core data on activity, amyloid, and tau. We will then explore the specific mechanism of tau mediation, discuss the dose-response relationship and its practical implications, and finally consider the study's limitations and directions for future research.
So, to begin, let's consider the preclinical AD puzzle. This early stage of the disease represents a critical window for potential intervention. Yet the precise mechanisms through which lifestyle factors like physical activity might alter the disease trajectory are still being defined. The potential impact here is substantial. As this quote from the Lancet Neurology indicates, a significant portion of Alzheimer's cases may be attributable to modifiable risk factors. Among these, physical inactivity is identified as a major contributor, particularly in North America and Europe. This brings us to the central research question. What is the specific biological mechanism by which physical activity might confer neuroprotection in preclinical AD? Is the effect primarily driven by an attenuation of amyloid beta pathology, a reduction in tau pathology, or does it operate through another pathway entirely? To investigate this question, the researchers utilized data from the Harvard Aging Brain Study, or HABS. This is a methodologically robust, longitudinal observational study of clinically normal older adults, making it well-suited to address these questions. The cohort included 296 participants with a mean baseline age of 72.3 years. A notable strength of the study is its duration, with a median cognitive follow-up of 9.2 years. This long observation period is essential for tracking the slow progression of preclinical Alzheimer's pathology. A key feature of this study is its reliance on objective measurement tools. Physical activity was quantified using pedometers rather than self-report. Amyloid and tau pathology were measured directly in the brain using specific PET imaging tracers. Finally, cognition and function were assessed using validated instruments like the PAC5 and the CDR sum of boxes.
Let's turn now to the results. The first relationship the researchers investigated was between physical activity and the primary Alzheimer's pathology, amyloid beta. Contrary to a common hypothesis, the study found no significant dissociation between baseline physical activity levels and the rate of longitudinal amyloid accumulation. This finding suggests that the neuroprotective effects of physical activity are likely mediated through a pathway other than amyloid prevention.
Where the data did reveal a clear signal was in a three-way interaction. Specifically, in individuals who already had an elevated amyloid burden at baseline, higher levels of physical activity were significantly associated with slower accumulation of tau pathology, slower cognitive decline, and slower functional decline. This effect was not observed in those with low amyloid burden.
This interaction pointed towards a potential mechanism. To test this, the researchers employed a moderated mediation analysis to determine if the effect of physical activity on cognitive outcomes was explained by its effect on tau pathology, specifically within the high amyloid group. The model tested a specific causal chain that in individuals with high amyloid, higher physical activity leads to slower tau accumulation. And this slower tau accumulation, in turn, results in attenuated cognitive and functional decline.
The results of the mediation analysis were quite clear. For cognitive decline, as measured by the PAC5, the model indicated that slower tau accumulation accounted for 84% of the protective association of physical activity. This suggests that tau is the primary mediator of this cognitive benefit.
For functional decline, however, the mediation was partial. Slower tau accumulation accounted for 40% of the relationship between physical activity and function. This indicates that while tau is an important factor, other mechanisms likely contribute to how physical activity preserves daily function.
So these findings lead to a practical question. What level of physical activity is required to achieve these protective effects? To address this, the researchers categorized participants into 4 groups based on their average daily step count: an inactive group, a low activity group, a moderate activity group, and an active group.
The analysis revealed a clear dose-response relationship. The inactive group showed the steepest cognitive decline. Moving to even the low activity level was associated with a substantial reduction in this decline. The benefit was greatest in the moderate activity group, after which the protective effect appeared to plateau. To translate this into clinical terms, the study estimated the time to meaningful functional decline for those with high amyloid. An individual in the inactive group was projected to reach this point in about 7.1 years. This was delayed to 10.2 years for the low activity group and 11.9 years for the moderate activity group, a substantial extension.
It's important to consider the study's limitations and how these findings inform future research.
First, because the design is observational, it cannot establish causality. It is possible that early neurodegeneration leads to lower activity, a concept known as reverse causality. Second, physical activity was only measured at baseline. And third, the cohort had limited diversity, which may restrict the generalizability of the findings.
Despite these limitations, the study provides valuable guidance for designing future clinical trials. The results suggest that prevention trials might maximize their power to detect an effect by specifically enrolling sedentary individuals who are also amyloid positive.
And perhaps one of the most practical takeaways is that the protective association appeared to plateau at a moderate level of activity. As the authors note, a goal of 5,000 to 7,500 steps per day may be more attainable for sedentary older adults than more aggressive targets.
The study's findings, while significant, also highlight remaining questions. Given that tau only partially mediated the effect on function, what other biological pathways could be involved? Future research will need to explore factors such as cerebrovascular health, neuroinflammation, and synaptic integrity to fully understand the remaining protective effect of physical activity.